I would first like to thank Stacy and her wonderful team for organizing this meeting. We are definitely having fun in your audience, and I'm sure the people on Zoom are also having a wonderful time. So thank you. My project, one of the major aim is to optimize pre-analytical and analytical conditions for qPCR telomere length measurements in whole blood, dry blood spots, and saliva. So these are the three main specimen types used in population studies of telomere research. For pre-analytical conditions, we propose to determine the best DNA extraction methods for these three specimens. We also wanted to develop quality control criteria for DNA samples, including DNA concentration, integrity, and OD 260/280 ratio and OD 260/230 ratios. We further would like to conduct a DNA stability study to test the effects of DNA storage condition, including storage time, concentration, and temperature. For analytical conditions, we propose to compare different commercial master mixes and different single copy gene primer sets. So Simone has talked a lot about the cross-lab DNA extraction methods for whole blood. For my presentation, I will be talking about some of the results we got from the DNA extraction methods for saliva samples. Idan had talked at length about DNA quality control criteria, and we, in our lab, we've conducted DNA integrity by running gels and measuring these 260/280 and 260/230 ratios. So we will definitely harmonize our study with Idan's ongoing studies. I will also present preliminary data on the DNA stability study. And finally, the third part will be our phase 1 results for the the master mix study. So the saliva samples we used for this study were collected in DNA Genotek's origin kits, which I understand is a very popular choice. This is actually a subset of samples from the National Growth and Health Study. We took 46 saliva samples that had been stored at room temperature for about 4.5 and 5 years when we conducted the study. These samples were extracted by 2 operators using 3 DNA extraction kits. So these kits represent the 3 main DNA extraction principles. At last year's meeting, I talked about the DNA quantification, qualification, quality assessments. So I'm not going to go through this year, but if there is interest to look at the data, I'm happy to share those with you. And today I will be focusing on the telomere length by qPCR data analysis. So first you see that there's a strong correlation between the 2 operators for the agent cord kit on the very left. And similarly, there's a very strong correlation between the 2 operators with the qAMP kit. However, if you look at the prep kit, which is a sorting out method, you see that the correlation between the 2 operators is only modest. We looked at the sources of telomere length variation, first stratified by extraction method. So as you can see, the majority of the telomere length variation comes from the participant. However, there is a much larger residual telomere length variation for the prep-it kit. So in this case, it's 17.87%. This is in agreement with the lower agreement coefficients between 2 operators for this specific kit. We did ANOVA analysis followed by pairwise comparison, and then we saw that there is a systematic difference between these 3 DNA extraction kits, with agent cord having the lowest T/S ratio and qAMP having the highest T/S ratio. So in the second model analysis, we further tested the telomere length variation source stratified by participant, operators, methods, and any interaction between any of the two components. So what I want to draw your attention is that we see a significant portion of variation in the participant by method. So what that means is that the difference between methods is participant specific. So our interpretation is that when you make an effort to systematically adjust T/S ratios in samples that were extracted by different methods, that does not account for this variation, which is participant specific. This is in agreement with the TRN's recent recommendation number 2, where we actually, based on the cross-lab whole DNA extraction experiment, we said that if there are cohorts or samples have been extracted with different methods, there is a way to analytically account for this variability, but it should be proceed with caution because this does not take into account the potential variability introduced by different DNA extraction methods. So the data I just presented with the saliva sample provide further support for this recommendation in saliva. So the second part of my talk is on the DNA stability study. This issue was already looked at in a paper published in 2017 by Dr. Gadola and her lab at NCI. So what they did is they extracted DNA with 3 DNA extraction methods in 10 subjects, resulting 30 DNA samples, and they compared qPCR results at the baseline versus DNA samples that were diluted at low concentration 1 nanogram per microliter and high concentration 20 nanogram per microliter, and stored at 2 different temperatures, 4 degree and minus 30 degree, for up to 6 months. So you can see that there's various degrees of correlation with the DNA stored at low concentration and low temperature, 1 nanogram, 4 degree, having the lowest correlation between the baseline and the 6-month data. And the next best would be the low concentration and low temperature, minus 30. And the high concentration, it looks like at 4 degree and minus 30, the correlations are pretty good, robust and good. But given what we now know about the importance of DNA extraction methods on qPCR, we as a whole team in the TRN felt it was important to further investigate this issue of DNA stability in the context of different DNA extraction methods. So the TRN DNA stability study actually is a collaboration between my lab, Aviv's lab, and Idan's lab. We took advantage of the cross-lab whole blood DNA extraction study that we were conducting at the time. So the baseline data actually was part of the data that Simon presented yesterday. So I'm presenting the data that actually just we just finished last week. My understanding is that Idan and Aviv's lab are still working on the samples, so I'm sure that we will get a lot more information when their data will be presented. So what we did in our lab is we took 20 DNA samples extracted by the Purging kit and another 20 extracted by the aging code kit. We had 2 different concentrations, 50 nanogram and 10 nanogram. And those were stored at 3 different temperatures, 4 degree, minus 20, and minus 80. These samples were stored as individual tubes at 3 months and 1 month. We measured qPCR. So here I'm showing a correlation heat map with the Purging kit. So what you see on your left is on the top of this heat map is baseline data, which was part of the TRN data set. And then we looked at these 6 different conditions. So from top to bottom, you can see the 3 different temperatures and high concentration, low concentrations in this order. So with the Purigen at 3 months, you can already see in pairwise comparisons, the Pearson correlation for some pairs were already not that great, as in pink and white. When we looked at 1 year sample, again, it's a separate aliquot, so it does not go through another round of freestyle. It's just a different aliquot. We saw that there were some correlations that became really low, and this is particularly true for this set at -20 with 10 nanogram per microliter, as you can see in red. We did also notice that when we run those samples on gels, there were a significant portion of samples that appeared to have no band on gels. And this is confirmed by the fact that on qPCR assay, when we look at the S reaction, the concentration measured by S reaction, which basically measures the genome copy, is also very low. And despite the fact that we started off all the samples with what we think is a fixed DNA concentration. So with that, we took out those samples and looked at the correlation map again. So in this, on the right side, when excluding those very low concentration samples, it looks like we were getting correlations that were very similar to the 3 month data. So this next slide is, again, the correlation heat map with the agent code kit. At 3 months and 1 year, you're not seeing a very obvious difference. So it looks like at both time points or pairwise comparisons, we have relatively robust correlations. If you look at each time point, compare Purigen and agent code, what we saw is that at 3 months, agent code seems to have more robust correlation. And of course, at 1 year, because we had these extremely low concentration samples for the Purigen, agent code does appear to be much better. Even when we took out those extremely low concentration samples, agent code still appeared to be better compared to Purigen at 1 year. So we did run all samples on agarose gels. I talked about how there were significant portion of samples that didn't really show a clear, obvious band, like we couldn't see anything. But for the samples where we do see clear bands, all samples seem to be intact. We could really detect any obvious degradation. So it seems that the poor correlation with the Purigen at 1 year when we exclude the low concentration samples was not due to any DNA integrity as far as we can see with our agarose gel imaging. So with that, I think we feel comfortable to say that compared to Purigen, agent code DNA is more robust under lower concentration and high temperature conditions at both 3 month and 1 year time points. And we're not really sure why Purigen is giving us low concentration samples, despite the fact that we dilute the samples at fixed concentration. I don't know if it's the DNA is completely degraded, or is we were having some troubles with getting a uniform concentration with Purigen samples, although the samples that we worked on were at relatively low concentration, 15 nanogram and 10 nanogram are not considered to be high concentration, but these are possibilities. And then finally, when we did see DNA on gels, it seems like there wasn't an apparent DNA degradation that we can see. So that's where we're at with the DNA stability study. This last part of my talk will be on the DNA master mixes. So we all know there are a lot of available commercial master mixes on the market. So in fact, it's just sometimes it's really hard to choose one because the ingredients are proprietary. So we don't really know how these master mixes perform until we actually try them. This is particularly true for the T reaction because by design for telomere reaction, the primers have mismatches to avoid primer dimers. So And you will see this, indeed, this is more problematic for the T reaction. And there are two previous studies that investigated impact of commercial master mixes on qPCR telomere length measurement. The first study was published in 2018, and they used human whole blood extracted by the sorting out method and tested 8 commercial master mixes. 4 are based on cyber green, and the another 4 are based on high resolution master mix, high resolution master mixes. They used qPCR, so basically it's a multiplex monochrome qPCR published by Richard Carlton in 2009, and I believe it's the same PCR machine that the Carlton lab uses. So this is table summarizes their findings, and I will just go through them point by point. So first of all, if you look at the first column, this is the crossing point for all the master mixes with the same sample, and you can already see there's a huge wide range with the crossing point with different master mixes. And the second column is the single copy gene. Similarly, you see a wide range of the CP values. Then if you look at the third column, this is the T/S ratio. So it's with the same sample, we would expect to see very similar T/S ratios, but clearly that's not the case. Although there are some master mixes that have similar T/S ratios, you can clearly see there's one with 0.63, the highest is 1.05. The other thing that's worth mentioning is that because it's a qPCR, by definition, the CP value for T should come up first in the reaction. So T reaction is expected to have a smaller CP compared to the S reaction, but that's clearly not the case for some master mixes. For example, if you look at the first line, T and S have a similar CP, so these two reactions are not really being differentiated with this master mix. Finally, my last point is that for some master mixes, there isn't a clear separation between the non-template control and the actual sample. For example, again, we look at the first master mix, the T with the sample is 19.1, and the T for the non-template control is 20.3. So clearly there's not a good separation. So I guess, you probably are convinced that not all master mixes are created equal. There's another paper that looked at, Saman, you'll like this. This is a bird qPCR, and they used qPCR to measure telomere length in 2 bird species, and they also tested synthetic oligos. And the assay platform they used is the 2002, which is a singleplex assay. They also tested 8 different commercial master mixes on the Quant Studio 3 platform. The results of this study is a little bit complicated, so I'm just going to try to summarize the key points. First of all, the CP values, again, are highly variable between different master mixes, so similar to the findings of the first paper I talked about, and that's true in both bird species as well as the synthetic oligo. And the second point is if you rank the CP values for these master mixes for the 2 bird species and for synthetic oligo, they don't agree with each other, meaning that if you have a high CP for this specific master mix in one bird species, you can end up having a lower CP in another bird species or in synthetic oligo. So there is not a correlation with the 2 bird species and the synthetic oligo. And finally, this paper also looked at amplification specificity from melting curves, and only 3 out of the 8 master mixes they tested gave consistent results with the 2 bird species. So this is, troubling for the T reaction, but that's clearly not the case for the S reaction because they saw across the whole all samples and all master mixes consistent CP or CT values for the single copy gene, and amplification is specific for based on melting curve and algorithm analysis for all samples on the S reaction. So the purpose of our current master mix study is to test different commercial master mixes on qPCR and being able to make recommendations. And I wanted to echo something that Simon had said in the past, naming that we did and we will test a lot of different master mixes, but the market is changing and there might be new ones on the market by the time, one is ready to do these assays. But I think just knowing the fact that different master mixes have huge impact on the qPCR assay is something we want to let people know, and we want to provide evidence to show that. And I think it's a point that we want to emphasize that master mixes need to be individually tested. So for this particular study, we plan to do it in 3 different phases. The first phase is just to test PCR efficiency and crossing point, and using a commercial genomic DNA as the reference standard. And the second, but this is really for screening purposes, and I'll show you some data. And the second phase is to compare TOS ratios of participant samples with master mixes that have gone through the predefined criteria in this first phase. And we really want to compare the TOS ratios with the master mixes to southern blot results. And finally, I would like to take advantage of the telomere clones that Lin's lab is developing. We talked a little bit about that in the past. So Lin's lab, in her talk, she talked about the telomere clones that she's developed. The beauty of using these clones is that the length of telomere sequence is fixed in these plasmid clones, and so this will get us to some accuracy issues. We can know exactly how many the base pairs and what are we measuring, the exact length of the sample we're measuring. So again, like I said, I'm only presenting the first phase data, which is the PCR efficiency crossing point of genomic DNA. So we started off with 7 commercial cybergreen master mixes based on major vendors. So these are the vendors I feel like I can trust in general. And it turns out one of them it was not available in USA. One of the master mixes is not available in USA when we looked. So we ended up with 6 master mixes, and we tested both the singleplex T reaction primers, so that's the Colton 2002 paper, and also the qPCR primer for T reaction. However, I want to point out that in our assay platform, we were actually using those primers on the singleplex. We did not mix the T and S reactions. So in this, we had identified 4 master mixes that passed our predefined criteria with PCR efficiency, and you'll see the data in a little bit. And with these 4 master mixes, we took them to the S reaction. In this case, we used beta globin as the single copy gene, and we tested 2 different PCR programs. One of them I call S reaction Lin 2010, with basically our standard, and that's the standard protocol that's on the TRN website. So that's what we typically use for our assays. And the second is the PCR program that's recommended by the vendor of that particular master mix. So with this S reaction experiment, we end up with 3 different master mixes. So this table just shows that the master mixes we tested, and as I mentioned, the market is changing all the time. Basically, the QIAGEN QuantiFast cybergreen PCR kit actually was discontinued after we finished that study, and it was replaced by a new one, and we haven't had a chance to test that yet. And I mentioned that we use a commercial human genomic DNA as the reference. So this just shows the standard curve concentration. So we did a 7.3-fold dilution, and so that gives us about 730-fold range. So these two graphs shows the T reaction PCR efficiencies. So on the left side is the 2002 primer set. I color-coded them to show that the blue ones are the ones that passed our criteria, which is efficiency between 85% to 105%. And also there's a clear separation between the non-template control and the lowest standard curve. So that needs to be greater than 5. And based on that, we have 3 master mixes that passed for the 2002 and the 3 master mixes that passed our criteria for the 2009 primer set. So these 2 graphs shows the CP values for the master mixes. Again, they're color-coded by the same as the previous slide with PCR efficiency and non-template control. So you do see there are some differences, but the difference is not as big for the 2002 primer set compared to the 2009 primer set. I think the one thing I noticed and I want to emphasize is that there's no correlation between the PCR efficiency and with how the CP value. So you could have poor PCR efficiency, but actually low CP value, meaning like early amplification. So the CP values were not used in our as a criteria. It's just for us to take a look. And this graph shows the S reaction PCR efficiency. For the S reaction, we used a little bit more strict criteria with the PCR efficiency between 95 to 100%. And again, similarly, we had asked the delta CP to be greater than 5 for the non-template control and the lowest concentration standard curve. And this is, again, looking at the crossing point. They do vary, but what we did notice is that with our standard single copy gene program, which is in SOCO, you'll see that it's always, they always have lower CP, so which means they always come up earlier in the amplification compared to vendor PCR program. So that's just something we noticed. So this slide basically summarizes the first phase of this master mix study. So we've identified 3 master mixes that with different combinations of T reaction primers and the S reaction PCR programs that we would take to the next phase. So this is shown in this slide. So the samples we plan to use is part of the next TRN cross-lab study that Simon had mentioned a little bit. So these will be PBMC samples and will be extracted with the qAMP. I'm not sure if it's MIDI or MAXI, but it's the same. It's the qAMP kit and will be extracted by one lab and distributed to different labs to measure on qPCR and the Southern blot. So we want to take advantage of this study and in addition, run the standard protocol in our lab. We would like to run the master mix, the 4 master mixes, 3 master mixes. So that's where I am at. I just wanted to again emphasize that the whole purpose of the master mix is that not only to identify master mixes that work at the current stage, but also to provide a framework of how in the future we want to test different master mixes that potentially could be coming in the market. So with that, I want to acknowledge the people who did the work and my funding sources. Thank you.
We have any questions? Oh, we have one.
Thank you. Thank you for all that work, the clear presentation. I know the standards are to have efficiencies within certain ranges, so it's quite reasonable what you're doing. But I wonder if low efficiencies actually cause, empirically, if we know that they actually cause problems, or if it's just kind of common knowledge that we do this as a rule of thumb, but without empirical basis.
So, so with our standard protocol, we typically get PCR efficiencies in the range of 85 to 90%. And honestly, I'm not seeing a very clear correlation between slightly lower efficiency and the quality of the data. So when we run, let's say when we run 2 runs, right? Occasionally you will get a run that has lower PCR efficiency compared to the next run of the same sample. And we don't really see a clear pattern that would lead to poor quality data. As long as it's not, in the very weird range of like 70 something, that's showing something is wrong with your reagent. And that happened.
Yeah, and part of the reason why I ask is I wonder, well, one of the things is we did some testing of master mixes for the multiplex assay, and I used to use the homebrew, and we recently switched to the power up because it was giving us as good or better results. And so it's interesting to see that it failed for you, but in terms of our repeatability, the power up actually we found to be better, at least better than our homebrew. The other thing that I'd suggest considering is you could look at the Lin-Reg PCR efficiency. So rather than using the standard curve, which can be dependent on the points on the standard curve and the sample that you're using in the standard curve, you can calculate the efficiency directly from the samples based on the empirical distribution. And Jue Lin and I have done that some, and it may be interesting if you want to dive into the efficiency and see how that good of a marker that is.
Yeah, we would actually love to try the Lin-Reg and see how that compared to. Yeah.
I believe we have someone raising their hand in the chat. Sung Pao.
Oh, yeah. Okay. Thank you for the talk. It's very interesting. One question is, one comment on that, for the DNA integrity that you use for showing the band that should be the high molecular weight to show that your DNA is kind of intact. But the problem is that if you are running the gel without separating them very well, sometimes you will still see the band on the higher molecular weight instead of that they might have a shift. So I would recommend you to look at the bands that is not only at a higher molecular weight. You have to compare them. Some of the higher molecular weight, they might have a lower, when they might migrate, they might separate differently. So for our experience, that some of those high molecular weight bands, actually, they are degraded compared to other samples. They are even higher molecular weight. So that is one thing that I want to comment on, that is because some people, they think they have the high molecular weight, they think there's no degradation. Actually, if they separate their DNA much better, you will see the difference. So that's the one thing that I want to comment on that. And one question I want to ask you is about the single copy genes. So it has been shown that different lab using different single copy genes. Some people use 36B, correct me if I'm wrong. And then you in your study, you use the beta globin. So will that affect the T over S ratio? If that affect, what's your comment on that? And one more thing, another question. I mean, one question is that seems like when you have the sorting out method to do your qPCR, you have less efficiency. My problem possibly will be either that sorting out is not have a good quality of the DNA, that might have some contamination, or it's because they preserve the DNA much better because they have a longer telomeres, because they can preserve the genomic DNA at a much higher molecular weight compared to the qIM. So that is one of the things that I'm thinking about. So I just want to know what your comment on that.
Okay, Sampo, I think there seems like 3 questions.
Yes.
DNA integrity. So what I did, okay, one kind of curious thing that I did not show in this data set is that with the saliva DNA, what we saw was really good agreement with 2 operators with qIMP, and also relatively good agreement between qIMP and AgentCourt. However, if you look at the qIMP DNA sample, you'll be horrified to see that there's clear degradation. So my sense is that
Actually, I'm not surprised.
Huh?
Actually, I'm not surprised about qIM will give you a degradation.
I can't hear. Say it again. I couldn't hear.
I'm sorry. Actually, I am not surprised qIM will give you the degradation because that's a membrane that will shear the genomic DNA.
Yeah, it's more so for saliva sample compared to blood. So we have gels for both sets of samples, and clearly for saliva you see a very clear degradation, but that does not prevent those samples to be at least consistent between operators and between AgentCourt, which has pretty intact DNA, and qIMP. So that's something that I wanted to point out. At least in our assay for platform, it doesn't have partial degradation. Doesn't seem to mean that it would perform worse on qPCR. Second, you talked about single copy gene. That's actually something that our lab is working on, and I did not present the data in this talk, but we have started this study, and again, it would go through like different phases. So we tested beta globin. We also tested RNSP. Based on PCR efficiency, they all worked very well. But what we did notice was a reagent lot difference when we tested 2 different lots. Consistently, one lot gave lower PCR efficiency, although they all passed above 95%. So that's in the next phase, we also would like to use the NCI samples to further test these different single copy gene primer sets. And then, sorry, I missed your 3rd question. Can you comment? Let's say again.
Yes. So I'm thinking about that, the sorting out that method you use that for the pure gene. It seems like it's less efficiency for qPCR from your lab. So my question is that possible because it's the purity of the DNA is not good enough for the qPCR, or is that preserve the longer genomic DNA compared to the qIM? So when you have the amplification efficiency, qIM has much better amplification.
We actually did not test the specifically PCR efficiency with different DNA extraction samples, but that's a very good point, and I think I would love to actually go back to the data and maybe use the Lin reg to look at the samples that extracted by different DNA and get an idea. So yeah, I'm not sure. So we did notice that I presented that with the 3D DNA extraction kits, it looks like overall there is systematic differences, right? The AgentCourt being the shortest, and qIMP, it has the highest T/S ratio. And I don't know if it could be due to the fact that qIMP is partially degraded, but it could also be carryover impurity from these kits, and I don't know which one.
Okay.
Hi.
we have-
Very interesting that you're comparing the master mixes on. Hello, can you hear me? I'm on the chat. So I was just interested in your comparison with the master mixes between on the Biorad machine. So were all the master mixes that you used, I'm not familiar with all of them, are they all like do they all have the same level of rocks? Because my understanding of the Biorad machine is that the optics are different than like other PCR platforms. Because I know we've tried to compare our PCR reaction on different platforms, and we've had to optimize primer concentrations to get good efficiencies depending on the platform and the master mix.
Yeah, so we used Roche light cycler, and based on what they tell us, having or not having rocks shouldn't make a difference. And I believe that some of them, maybe one or two, I can't remember, has rocks, but our machine, based on what the vendor says, should not matter. Yeah.
Thank you.
Do we have any more questions in person or on the chat?
Okay, well, with that, we are going